Hi, I'm Marlene Mika and I'm going to continue the topic about microbial virulence factors and mechanisms. So, aside from the bacterial structures such as fimbriae or pili, flagella, and capsule, another important virulence factors are the exoenzymes and toxins. So, in exoenzymes, this includes the necrotizing enzymes, coagulase, kinases, hyaluronidase, collagenase, hemolysins, and lecithinase. And in toxins, there are two major categories which are the endotoxins and exotoxins. And there are also two types of exotoxins which are the neurotoxins and enterotoxins. So let's discuss them one by one. So first, let's discuss what is exoenzymes. So what is exoenzymes? So when we say exoenzymes, it is an important virulence factors that pathogens produce. So some pathogens release enzymes called the exoenzymes that enables to evade host defense mechanisms, to invade and to damage host tissues or body tissues. So in in exoenzymes, this includes the necrotizing enzymes, number one. So in necrotizing enzymes, it is a virulence factors that cause the destruction of cells and tissues. So notorious examples are the flesh-eating strains, Staphylococcus pyogenes that produce proteases and other enzymes that cause a rapidly destruction of soft tissues. So, um, leading to a disease called necrotizing fasciitis. Number two, coagulase. When we say coagulase, it is an exoenzymes that cause clotting. So, it is an important identifying feature of Streptococcus aureus in the laboratory because it has an ability to produce a protein. So, coagulase binds to prothrombin, forming a complex called staphylothrombin. So, the protease activity of this thrombin is activated on this complex, causing a conversion of fibrinogen to fibrin. So, in the body, coagulase may enable Streptococcus aureus to clot plasma, thereby forming a sticky coat of fibrin around themselves by for protection from the phagocytes, antibodies, and host defense mechanisms. So that is coagulase. Number three, kinases. When we say kinases, it is an exoenzymes that dissolve clots. So it is also known as fibrinolysins and have the opposite effect of coagulase. So kinases dissolve clots. Therefore, pathogens that produce kinases may enable to escape from clots. So there are two enzymes that includes in kinases. One is the streptokinase, a kinase that produced by streptococci. And number two is staphylokinase, a kinase that produced by staphylococci. So, because Streptococcus aureus can produce both coagulase and hyaluronidase, or staphylokinase rather, um, not only Streptococcus aureus can cause the formation of clots, but it can also dissolve them. Number four is hyaluronidase. When we say hyaluronidase, it is the spreading factors because it enables pathogens to spread through connective tissue by breaking down hyaluronic acid. And the polysaccharide cement hold the tissue cells together. So hyaluronidase is secreted by several pathogenic species of Staphylococcus, Streptococcus, and Clostridium. Number five, collagenase. When we say collagenase, it is produced by certain pathogens and it breaks down collagen. So collagen is the supportive protein found in tendons, bones, and cartilage. So this enables pathogens to invade host tissues. So Clostridium perfringens, a major cause of gas gangrene, spreads deeply within the body by secreting both coagulase and hyaluronidase. Number 6, hemolysins. When we say hemolysins, it is virulence factors that cause damage to the host red blood cells or erythrocytes. So, not only does the lysis, bursting or destruction of red blood cells to harm the host, but it can also provide the pathogen with a source of iron. So, that is hemolysins. Number 7, lecithinase. When we say lecithinase, it is the virulence factors that cause the destruction of host cell membrane. Clostridium perfringens, a major cause of gas gangrene, is able to rapidly destroy the extensive areas of tissues, especially the muscle tissue. So, one of the enzymes produced by Clostridium perfringens is lecithinase. It breaks down phospholipids, collectively referred to as lecithin. So this enzyme is destructive to the cell membrane of red blood cells and to the other tissues. So let's move on to the another important virulence factors, which is the toxins. So what is toxins? When we say toxins, it is the ability of pathogen to damage host tissues and can cause disease. May depend on the production and release of several types of poisonous substance. So there are two major categories of toxins. Number one is the endotoxin. So when we say endotoxin, it is the component of the cell walls of gram-negative bacteria. The cell wall of gram-negative bacteria contain lipopolysaccharide, and the lipid portion of which is called the lipid A, or what we call the endotoxin. So endotoxin can cause serious adverse and physiologic effects such as fever and shock. So substances that cause fever is called pyrogens, while the type of shock that results from the gram-negative sepsis is called the septic shock. So that is endotoxin. Number 2, exotoxin. When we say exotoxin, it is a poisonous proteins secreted by a variety of pathogen, and they are often named for the target organ they affect. So there are two types of exotoxin. Number one is neurotoxin. So what is neurotoxins? So when we say neurotoxins, it is an exotoxins that adversely affect the central nervous system. So Clostridium tetani and Clostridium botulinum, tetanoplasmin and botulinal toxin can cause tetano and botulism. So in tetanoplasmin, it affects the control of nerve transmission, leading to a spastic, a type of rigid paralysis in which patients' muscles are contracted. And in botulinal toxin, it also blocks nerve impulse but by a different mechanisms, leading to a generalized flaccid type of paralysis in which patients' muscles are relaxed. So that is neurotoxin. Number 2 is enterotoxins. So what is enterotoxins? So when we say enterotoxins, it affects the gastrointestinal tract, often causing diarrhea and sometimes vomiting. So examples of pathogenic bacterial produced by enterotoxins are the serotypes of E. coli, Bacillus cereus, Clostridium perfringens, Clostridium difficile, and many more. So in addition to the releasing of enterotoxin, or called the toxin A, Clostridium difficile produces cytotoxin, or what we call the toxin B. to damage the lining of colon, leading to a condition called the pseudomembranous colitis. So that is enterotoxins. So those are the microbial virulence factors and mechanisms. Thank you.